My name is Frederic Bushman, and the lecture today will be on the composition and dynamics of the human virome. We will be considering not just viruses, single viruses that infect people, but the large viral communities, the huge viral communities that live associated with the bodies of each human. So the introduction, what is the human virome, some sort of overview findings that help us understand its nature. Then targeted hypervariation. It turns out some viruses mutate themselves at high rates to accelerate their evolution, and we'll learn more about that. And lastly, we'll investigate the question, why do different humans harbor such different viral communities from one individual to the next? So the global virome is remarkable. There's in a milliliter of seawater in the world's oceans, there's something like 10 to the 7th viruses, 10 million viruses, absolutely enormous amounts. Viruses outnumber their hosts by something like 10-fold in seawater. This is also seen in freshwater lakes, and as I'll be saying, we think viruses outnumber bacteria in the human gut. There are something like, estimated to be something like 10 to the 31 viral particles on Earth. So numerically, they're the most successful biological entities. You could say that the story of the biosphere on Earth is the story of viruses and a little bit of other stuff. And then in the lower right corner here are some pictures of marine viruses taken by Lita Proctor. Okay, the human virome is similarly gigantic. People will commonly be familiar with the persistent or latent infections that most of us harbor. So many of the herpes virus, Epstein-Barr virus, herpes simplex infect most of the population. Papillomaviruses also. Unfortunately, HIV and HCV also infect a substantial fraction of the world's population. We're also transiently infected with viruses, like the cold virus I have now. Sometimes we deliberately infect ourselves with viruses, as with vaccinia, which is the vaccine strain for smallpox that was used in the smallpox eradication effort. But still, that's really just the tip of the iceberg. The human genome itself is composed of 8% discoverable viral remnants, the so-called endogenous retroviruses. So what's shown here is a bit of a human chromosome, chromosome 11. The genes are shown here. And I've asked the browser to show the human repeated sequences, which are shown here. And this track labeled LTR means long terminal repeats. Those are the structures at the ends of retroviruses. And you can see there are many, many ticks as you go along this genomic region. There's a large number of fragments that are identifiable as remnants of integrated retroviruses. So 8% of the human genome is discoverably produced by viruses. And then another giant community of viruses associated with humans are the bacteriophages, the viruses that prey on bacteria and archaea, on the very dense communities that live in our guts. Our guts have something like 10 to the 11th bacteria per gram, and there's even more bacteriophages, we think, in the human gut. So thanks to the new DNA sequencing methods, it's possible to study viruses using these amazing new methods. So you can hunt for pathogens, you can watch viral evolution playing out by sequencing longitudinal samples and observing the sequence variation that happens. If you're interested, you can characterize the sites of integration by human endogenous retroviruses or other retroviruses into different genomes, and try to understand the biological ramifications of that, such as HIV integration. And you can characterize complex uncultured communities, try to understand their membership, and try to understand how they change over time. And this last point is going to be the main theme of the lecture today. And so this is all supercharged by the amazing new DNA sequencing methods. You can literally get more than 100 billion bases of sequence information from a single instrument run using the new amazing Illumina HiSeq instruments. And so we and many others have been studying viral communities using the sequencing methods to report the viruses that are there and using that to try to understand their nature. So viruses of the human gut, their particle counts show something like 10 to the 11th viruses per gram of human stool. And with sort of more advanced nucleic acid-based methods, that's what we typically see. Sequence analysis shows most reads correspond to novel sequences, not sequences that are present in databases now. And that's not surprising because remember I said that there's these enormous populations of phages in the world, 10 to the 7th per mil in seawater. There are only a few thousand in databases, so it's not surprising that if you capture a new phage in the wild and sequence it, that it's generally not going to be close to something that's already there in the databases. So if you sequence some phage from a human, for example, typically what you'll see are sequences that don't closely resemble any sequences that you've seen before. And then, again, because of these gigantic population sizes, if you sample several different humans, you get several different communities that show very little resemblance between individuals. So the kinds of things we want to try to figure out: what viruses comprise these huge populations, what's the basis of this interpersonal difference, and how are these communities evolving and changing over time. And again, many strong labs have contributed to understanding the human virome, some listed below there. So just to give you an overview of some of the kind of data we get, here's some summaries from a paper we published a couple years ago now. So we sequenced 40 billion bases of highly purified viruses from 12 individuals using the Illumina HiSeq method. So what we did, we took human poop, carefully filtered, treated fractions with nucleases to remove free nucleic acid, and then broke open viral particles and sequenced. And a bunch of control experiments tell us that most of what we had at the end of that purification procedure was in fact viral particles, so high purity starting material. So we then assemble into contigs. That is, we take short sequence reads and merge them into longer assemblies, which we nicknamed contigs. So the upper left here shows the contig spectrum, where we have length on the x-axis of these assemblies going out to 100,000 bases, and sequencing depth on the y-axis. Some bases were covered up to 1,000 times at each position. So you can see we've got lots and lots of different viruses or pieces of viruses. Some of these shown in red are circular, and so we guess that these are ones that we completed the sequence on, because many viruses are known to be circular or circularly permuted, so that we think we caught everything. The other ones, the linear ones, some are probably linear viruses, others fragments of viral genomes. So if we annotate the genes that are there in part B here, each column is a different subject, and the kinds of genes are shown in the color code on the right, and the proportion of annotated bases is shown. Most bases don't annotate, but of those that do, we get stuff that looks like you'd expect for viruses: heads, tails, integrases, genes important for regulation, and then an interesting collection of further genes like restriction enzymes, antibiotic resistance genes, and reverse transcriptases, which I'll tell you more about a little further along. So most sequences didn't look like anything we've seen before. The individuals showed very little resemblance to each other, but again, we think this is a consequence of the giant population size of the viruses that we're studying. In C here, we show alignments to databases. In one case, this human papillomavirus, we got very good coverage. The gray shows coverage in this kind of a picture, and blue shows some papillomavirus genes underneath. The red streaks show a mismatch to the database virus, the colored streaks show a mismatch to the database virus. So you can see we covered this small virus. It's human papilloma virus type 6b is sort of vanilla, well-known kind of papillomavirus. And this kind of helps us believe that our methods are working properly. However, when we align to our other alignments, now to bacteriophages, show very sketchy matches. You can see how patchy this is. This is taking our reads and aligning against databases. Blue shows the genes called in the databases. And we get very sketchy alignments with lots of colored bars indicating mismatches. And these were our best from the whole collection. So what this is saying is that most of the viruses in our collection, the viruses that we see in A here, do not closely match what's present in databases. Okay, So if we work hard, we can do better understanding or identifying genes in our viral collection that are present in the databases and try to understand more about them. So only 25% of the phage open reading frames, that is inferred genes, in our data set have matches to database open reading frames using a relatively permissive threshold. So we compared genes for similarity within the data set over this the 40 billion bases of sequence, this very large number of contigs. And we do a little better, actually, comparing within the data set, just because it's so large. We found that 58% of genes had at least one match at that level of 30% identity, a permissive threshold. So one thing we wanted to investigate was whether these genes are coming in cassettes. It's well known in phage that often there'll be blocks of genes for specific functions, like heads, tails, replication, what have you. So we tried to investigate that within this data set. We looked for conservation of gene type and order and orientation. And we found 28 cassettes containing from 2 to 8 protein coding families. And so shown here is one of our most spectacular, where we've colored genes by their resemblance. And you can see we can find sets of genes that look like they're traveling together in this phage collection. And you can see they come from multiple different subjects. So this looks like a conserved cassette across the data set. Looks like something that was seen previously in mu-F, in phage mu, F gene, for at least one of these cassettes. So we found that contigs were covered by something like 27% by cassettes. So again, if we try hard, we can start to see some patterns in the genes that we're recovering and some resemblance virus by virus, but mostly thanks to comparing within this very large data set. Again, the database is being relatively unpopulated. So we can also examine gene content in the bacterial community versus viral communities, and that was kind of fun. It indicates that viruses are parasites, as is of course very well known. So each pair of bars here indicates a kind of a class of genes, and we're comparing whole stool sequence, the bacteria in orange, to viruses in red, and the relative proportions of each in each community. So you can see that the bacteria devote a lot of their coding capacity to amino acid metabolism, carbohydrate metabolism, translation. Viruses do little or none of this. Instead, they devote much of their coding capacity to RNA processing, to replication, recombination, and repair, and also to functions we don't know what they are. So this really says that viruses are stealing macromolecular precursors from their hosts in order to promote their own replication. This is, of course, very well known from many, many kinds of studies in virology, but it's cool to see it playing out in the metagenomic data presented here. So I've been emphasizing that in the gut, in healthy people, we overwhelmingly see bacteriophages. Now, that's not always the case in disease states. It's very well known, of course, that viral infections cause many different diseases. So here's just an example of picking out a little-known virus in a disease state. So we studied 3 kids, stool samples. 2 of the children suffered from severe combined immune deficiency, a very severe immune defect. And we took stool and did metagenomic sequencing. And we found for one of these kids, 20% of the sequence reads, a very high fraction of all the sequence reads, corresponded to bocavirus. Now, bocavirus is a little studied parvovirus. It was only discovered in 1995. You wouldn't even know it existed at all if not for the metagenomic sequencing methods. This picture shows the genome and just the coverage. Each gray bar indicates a sequence read. So there's some hypervariable regions that are pretty cool. Here's a picture of another parvovirus, just to give you an idea. It's a spherical DNA, non-enveloped DNA virus. So we got very large amounts of this virus in one kid. And so this is just an example of many, many studies showing that in healthy humans, there's not a lot of animal cell viruses, DNA viruses in stool, but in disease states, of course, you can find a lot more. So importance of bacterial viruses, although they don't cause direct don't directly attack human cells, they do influence human health. They define the structure of bacterial populations by predation. And importantly, they can transfer new genes between bacteria, install those genes into host cells by lysogenization, that is integration of the bacterial DNA into of the viral DNA into bacteria. So, and then those genes are transmitted just as regular old bacterial genes. And so the kinds of genes that are installed by lysogenization can have an important effect on the phenotype of the bacterial host. For example, Shiga toxin, cholera toxin are carried on phage, and whether or not they're integrated in a bacterial genome will have a big effect on how toxic colonization with that bacteria is. Viruses, phages also carry around virulence determinants, genes for antibiotic resistance, genes that change the metabolic capacity of bacteria. So by all of these means, bacterial viruses can influence the function of bacterial cells and thereby the degree to which those bacterial cells influence the condition of the human host. So that's some overview on the human virome. We will now do a study of variation in these virus populations, targeted hypervariation. So Sam Minot, the talented student who did this work, having studied these bacterial genomes in depth, wanted to ask, well, in these populations, are there regions that are particularly variable at the sequence level? This is of interest because in HIV, for example, the surface proteins that are exposed to the immune system will change rapidly. So there's reason to think that genes that are under pressure to change rapidly, so the proteins they encode can evolve rapidly, may be particularly important for function. So digging into this sort of giant collection of gut viruses, Sam asked, well, where do we see hypervariation? So in this upper left part here, you can see a region of a contig. The gray shows the coverage map, and there are a bunch of genes in this region. That's the blue, some of which are identifiable. Now, if you blow up that region here, you can see there's a bit that is where there's lots of colored bars indicating mismatches. In other words, a quite variable region. And so that's shown down here, lower left. Each column is a base, and it's colored by which base it is. And each of these lines is a sequence read. And so this is a gap between sequence reads, this white bit here. So what you can see is there's lots and lots of substitutions, lots of different colors as you go from read to read. In fact, this region is so variable that it's almost the case that every read is different from every other read. And that's extremely rare in these genomes. Most of the time you see, as in this bit up here, all the columns are all the same, indicating consensus that there's a single sequence there. Now these regions had some character, which was really cool. There's a sequence and a matching sequence nearby, so it's duplicated, and then there's a reverse transcriptase gene nearby. This reverse transcriptase, of course, copies RNA into DNA. So what on earth is the deal with that? We see these 100 base regions, where nearly every read is different from every other read, and they're associated with repeated regions and reverse transcriptases. Well, it turns out that there's been some really beautiful work done on one of these kinds of regions by Jeffrey F. Miller and coworkers. And so I'm going to tell you about their mechanistic studies and then come back to the kinds of things we saw in the metagenomic data. So Miller studied phage BPP1, which infects Bordetella. Now BPP1 has a problem, which is that Bordetella, the bacteria, will change its surface coat periodically, going from red to blue here, in response to immune pressure. It undergoes phase variation, and so the bacteriophage can no longer bind. So that's bad if you're a bacteriophage. But it turns out what the phage does is it causes hypermutagenesis. It varies this tail protein that contacts the bacteria. It's called the MTD, or major tropism determinant. It binds, so the protein that binds bacteria is also changing rapidly. And in fact, when the bacteria undergoes phase variation, there's commonly a variant in the population, a phage variant that can bind, and so proliferate and continue the infection. So the mechanism has been really beautifully worked out by Miller and his colleagues. So here's the gene for the major tropism determinant, and it has this variable region near the region encoding the C terminus. Now it turns out information moves from this template region to this variable region by means of transcription of the template region, reverse transcription in an error-prone fashion, and incorporation at this site. And that's how hypermutagenesis takes place. Miller showed that if you get rid of this template region, it doesn't work anymore. Get rid of reverse transcriptase, it doesn't work anymore. If you mutate, if you insert a self-splicing intron into the template region, you can see information move from the template region to the variable region with clean removal of the intron, splicing out of the intron, showing conclusively that it went through an RNA intermediate. Information went through an RNA intermediate going from the template region to the variable region. A beautiful demonstration of the role of reverse transcription. So we see regions that look like Miller's major tropism determinant. We also see other kinds of genes being targeted for hypervariation. This is an example of an immunoglobulin superfamily gene, which is targeted by this same system, reverse transcriptase and template region. And that's really cool. It's an example of convergent evolution. The human immune system, of course, the T-cell receptor and antibodies, the genes encoding each of those, are immunoglobulin superfold family members that are mutagenized, but in that case by the RAG recombinase system. Here it's targeted hypermutagenesis by the diversity-generating retroelements, reverse transcriptase-dependent mechanism. It's been argued for antibodies and for the T-cell receptor that the 7-strand beta barrel fold, characteristic of the Ig domain, is particularly stable, and that's good for a scaffold that's getting mutagenized because it preserves the fold despite the change in amino acid sequence, and so maybe some of the same logic holds here. So that's really cool. We're picking out new kinds of genes that are subjected to this hypervariation mechanism. So there's a specific targeted sequence, AAY, for this hypermutagenesis mechanism, which makes perfect sense in terms of the genetic code. So AAY is an asparagine codon, and the A's in AAY are specifically targeted. Here's the variable region, target region. You can see A's getting targeted of the 4 possibilities. So the reason that makes perfect sense, if you mutate an A in AAY, you can go to any of the other amino acid side chains, chemistries, that is positively charged, negatively charged, bulky hydrophobic, hydrophilic, but you can't get to a stop codon in a single step. So it makes perfect sense that this would be targeted if you're trying to make a protein with changed binding properties, but still preserve, but not killing off the protein by introducing a stop codon. So we aligned the reverse transcriptases we saw to databases of reverse transcriptase genes. That's shown here. And what we see is that a specific clade tends to be the most populous in phage associated with these diversity-generating retroelements. Jeffrey Miller's example is here, and Virome, our Virome sequences, and green shows those that are associated with hypervariation. All of them are in this branch of the reverse transcriptase family. The retroviruses are over here. So what that says is that in bacteriophage, in DNA phage, the main job of reverse transcriptase seems to be this targeted hypervariation. It's where we see by far the most RTs showing up in this branch that seems to be devoted to diversity-generating retroelements. And so lastly, some speculations about the role of those poly-Ig domain proteins that I mentioned. So we don't know what they do, but there's an example of one of these poly-Ig proteins that it has been studied pretty carefully in bacteriophage T4. And although it's not hypermutagenized, we can make some speculations about what the hypervariabilized proteins do biologically. So this T4 protein is called HOC, structure shown here on the left. HOC, it binds to 6-fold centers of symmetry in the bacteriophage head, shown in yellow here. So there's over 100 copies of HOC on a T4 head. So it's a dispensable phage protein. people have done bioengineering to put ligands on here and shown that that can change how the phage particle binds to stuff. So the idea would be that the phage is engineering its binding specificity in a diversifying way that then can be a substrate for Darwinian selection. So if you introduce a single new positive amino acid onto Hock, you get over 100 new positive charges on the surface. So that would greatly change the binding properties of the phage. So Hock has been suggested to be important in receptor binding on cell surfaces. So maybe it probably is partly devoted to binding target cells. But there's no reason it has to be restricted to that. In the environment, who knows what specificity is going to be most important? Maybe binding to a mineral surface or a passing kelp leaf or whatever could make the virus more fit, allow it to leave more offspring in the next generation. So you can see the phage accelerating its evolution of its binding specificity that can then be a substrate for selection in subsequent rounds. So we think it's sort of exciting to see that this diversity-generating retroelement system may be targeting proteins like this to change how the phage binds to in the environment. Okay. So that's a section on targeted hypervariation. Phage are accelerating the evolution of binding specificities in ways that look like they matter to them. So why do humans harbor such different viral communities? So here we carried out a study of longitudinal variation in phage, where we took samples from one individual over 2.5 years and asked how the communities were changing over time. So we purified DNA viruses from sampling scheme shown here. At some time points we sampled twice, so we could compare within time point variation to between time point variation. 57 billion bases of sequence information from Illumina HiSeq assembled. We also did shotgun sequencing on total stool DNA to know what the bacteria were doing. We got something like close to 500 viral contigs with an average of 82-fold sequence coverage. And so with this dense sample set, we can ask, well, how are these populations changing over time? So here again is a contig spectrum. Here's contig length on the x-axis by fold coverage on the y-axis. You can see for some of these we're now up to million-fold coverage, so we've extremely clearly figured out some of these genomes. Blue shows circular, so you can see many of these now are circular, indicating that we completely caught them. Some are linear, indicating either that we got linear viral genomes or that we only have pieces of genomes. We asked how the community membership changed over time. Here's longer and longer time intervals between sampling points, and here's the Jacquard index, which indicates shared membership. And so what we find is that at our longest time points, something like 80% of the community members are the same. So that indicates that mostly the viruses were hanging around, although our measurements indicate they were changing in abundance, but presence absence, 80% are still there. We could also ask if we'd saturated our sampling, and you can see as we add more reads, we flatten out in the number of contigs that we catch. So that's saying that we're close to saturation, at least with this method, though we do know with other methods we can catch some different ones if we try hard. So 60 circular assemblies suggestive of completed sequences. Most lineages persist over 2.5 years, and rarefaction suggests saturation by this method. So one thing we asked is, can we see diversity generating retroelements, and are they active in the samples from this one individual? And the answer is yes. We could compare. We found 6 different ones, and then we could compare between time points and ask, do we see them changing? Can we obtain evidence that they're active inside this individual that we're studying? And for one, we were able to find that yes, they were changing over time detectably, although for the other 5, we don't know if they weren't changing or whether we were underpowered to detect them, and that was the reason we couldn't find activity. However, it does raise an interesting question. Might the DGR, diversity generating retroelement system, be regulated and turned on at times when it's needed and kept off when it's not? So that's an interesting problem for future research. We also asked about the CRISPR system. Very interesting new area, probably familiar to many of you, but I'll introduce the CRISPR system on this slide and then tell you about what we found in another slide. So CRISPR stands for clusters of regularly interspaced short palindromic repeats. It's a form of bacterial adaptive immunity discovered just in the last few years. So the way this works, a virus infects a cell, introduces DNA. Some of the time the DNA is chopped to remove a piece that's incorporated into these so-called CRISPR arrays. DNA in these arrays are transcribed to make RNAs, which are then chopped up and used and bound by proteins and used as recognition elements in a nuclease complex, so that if the blue DNA comes in again and there's a CRISPR spacer bound by the targeting nuclease, it will bind and chop the incoming DNA, protecting the cell from viral infection. And so other forms of mobile DNA can also be chopped, including plasmids and other forms of invading DNA. So remarkable new discovery. We could see action of the CRISPR system in our data set. So remember I said we sequenced bacterial DNA also from stool from this individual. And so we found CRISPR systems in what we infer the bacteria. And we found 6 viral contigs that were targeted by bacterial CRISPRs, and as many as 27 repeats targeting a single viral contig in a CRISPR. And so this just shows one of our contigs, and arrows show the points where CRISPRs could be found, bacterial encoded CRISPR spacers matched to sequences in the virus as shown here. We found one example of a possible viral escape mutant. So x-axis shows time, y-axis shows relative abundance of contig encoding different sequence variants. So a contig hangs around for a while and then falls in abundance and stays low. And what comes up is a new variant that has a mutation in a sequence that matches a CRISPR targeting sequence. So this would be consistent with the idea that this point mutation allowed the virus to escape the pressure exerted by that CRISPR spacer. We haven't tested further. We don't know if that's really the case, but this is certainly what you'd expect escape to look like. And then we had some interesting further findings. So we could find CRISPR systems on bacteriophage, and others have actually commented on this also. We could find CRISPR spacers on phage, and in one case we found a CRISPR spacer on one of our phage that was targeting another phage in the same individual. So it turns out that, so this is evidence that phages are fighting it out with each other using the CRISPR system. Phage encode many functions to fight with other phage. They tend to, in many environments, they outnumber their hosts. And so we think the CRISPR system has been recruited for interphage combat. Another form of variation we studied was just accumulation of point mutations. One thing we found was that temperate phages tend to have lower values. So temperate phages are ones that can form lysogens, as I mentioned earlier. So phage can infect, introduce its DNA into a bacterial cell, and a temperate phage can then integrate that DNA into a host cell to form a lysogen, as shown here. And then under unfavorable circumstances, it can induce and burst the cell and produce progeny. We found that phages that can do this temperate thing seem to evolve more slowly than phages that are not temperate, at least for the ones that we could assign and study by this means. Many of the phage we couldn't recognize, but by a few different criteria we could sort of guess what family some of these phages were in and thereby annotate whether they were temperate or not. And the temperate ones showed fewer substitutions per base per day. Other families seem to evolve rapidly, and our champions were the Microviridae, which is the group that includes phage Phi-X174. These are single-stranded circular DNA phages. Single-stranded DNA viruses tend to evolve faster than double-stranded, presumably because if there's a change in one base, there's no template to correct it against. So these here we see time, our sequences shown as the bars, and time going from top to bottom. And you can see accumulation of colored point substitutions in each of these microviridae that we found. Here, this shows a phylogenetic tree showing that these guys have unique placements, and it's not new ones coming and going. So our champion here was one that showed 4% substitution over time, correcting for within sample variation, which we also measured by the repeated sampling scheme that I mentioned. So what's cool about that, some of the microviridae species in the viral taxonomic database are separated by as little as 3.1% substitution. We saw 4% substitution accumulate in one virus over 2.5 years. So that's coming into the range where we could proclaim this a speciation event. Now that's a little forced. What a virus species is open to some debate, but we can say we're seeing levels of change in some of these viruses that you'd at least begin to consider calling viral speciation in just 2.5 years. So it's absolutely remarkable. It raises the idea that maybe inside each of us, new kinds of virus evolve during our lifetimes. So why are humans so different from each other? Well, bacteria in their viral populations, bacteria, the bacterial microbiome, I haven't emphasized this, but one of the main findings in all of the recent microbiome projects has been that humans are really different from each other at each of the body sites sampled, particularly gut. So the fact that we have different bacteria in our gut, individual by individual, and phages are predators on those bacteria, presumably will help make it so that we'll each have different phages as well, preying on the different bacteria that we house. But so that's probably almost certainly part of it. But we think there's another piece here, which we found in the study I just described, which is that at least some of the phage are changing really, really fast. And we can specify some of the mechanisms: steady substitution, as in the microviridae, diversity generating retroelements, the RT targeted mutagenesis, response to CRISPRs, and other means too. So that concludes what I wanted to say. I introduced what the human virome is, overview of its enormous size and scope. We are made out of viruses, 8% of our genomes, and there probably are more viruses living associated with our bodies than there are bacteria. When we look at how they change, we find this remarkable targeted hypervariation, reverse transcriptase, targeting specific sequences to change, probably altering the binding properties of the viral particle. And then why do humans harbor such different viral communities? Well, we harbor different bacteria, and so the predators, the phage that grow on them, will differ between individuals. But also, some of the viruses are changing really, really fast, so that we probably evolve our own kinds of viruses during our lifetimes, at least for a few of them. And so let me just credit my colleagues. Sam Minot did a lot of the work that I described. Alexandra Bryson, Emily Charlson, and others have also contributed. And my co-PIs on microbiome projects are Gary Wu, James Lewis, Ron Coleman, and work with a terrific group of collaborators. Much of the work that I described was funded by the Human Microbiome Project. We were a demonstration project, and also these other players. So thank you very much for your attention.